a few more questions before we turn it over to our audience. And Eric, I'll start with you on this one. Can the phenotypic effects of genome editing can they be accurately predicted? Can you talk us through that?
Yeah, and so I think people, organizations have targeted so far things where are diseases where the phenotypic effects are closely linked to the genotype, and so it's more predictable. Obviously, there's many complex diseases out there that have multiple risk factors, might be multigenic, polygenic in nature. And I think that, that's where it comes in. Well, that's where you have to really define the disease pathway upfront in preclinical studies, and how the protein or the gene and ultimately protein you're targeting is how it's related to that pathway. Accenture Labs is doing some work in this space as well for understanding how when gene are off-target effects, but also how that gene is related to the protein the expression pathway and overall biological effect in the cell. And so we can look at how those proteins interact and hopefully better characterize not only what happens if you have an off-target effect, but how does that off-target effect affect overall gene expression and ultimately the biological properties of that cell. So I do think that's one point that needs to be considered, is it's not just about, oh, I didn't target the correct spot, but what's the overall impact of an off-target effect biologically on the cell, so you can fully understand the off-target.
Okay, great. Thank you so much. Steve, I have a question for you. I know there's a very big regulatory component to what I'm about to ask, which you could probably do a whole other event on. But my question is, should we edit the human germline? Why are we not? And, what are the opportunities? What are we looking at?
Well, as we all know, it happened, right? And there were actually 3 babies that were born in China that had genome edits. And I think that, well, there was a moratorium then instituted, although even now there are people that are using genome editing in, say, multi-cell embryonic cells, at the multi-cell stage, but not beyond that. And I think that really the issue comes down to everything that we've been talking about in this little discussion here, and that is that we really don't understand what the tool does. And so whichever tool would be implemented for germline editing, you have to understand what its capabilities are. And so even if you think you're only getting the on-target change, and if that was even true, you don't know what the effects are as an individual, matures and then is challenged, right? And so that, I think, was one of the big questions about the changes that were made in the babies that were born, is what if they're challenged by some virus or something like that as they mature, and their immune system is modified by that change in a way that we don't understand, and what is their ability to, say, fight that virus? And that's really, the big question is, what do these modifications do as you're challenged in terms of development? Because you're very different now than you were when you were an infant. And those changes could be challenging via the change that was made. Then there's all the off targets, right? And so the next level down are the off targets that you can find. And then the next level down from there are the off targets that you don't even know about, right? And so as we were talking about earlier in this call, you could be looking for off targets that are identified through your tools, whatever tool that is, and say, well, that was in an intron and it's not going to have any impact or something like that. But there's all the ones you don't know about when you just made like a 10 kb deletion somewhere in the chromosome that you didn't even look for. And so this is where the problem comes in with germline editing, is that we don't know the capability of the tool, and it really boils down to the sequence that you use to create the genome edit of interest and what the off-target and off-tissue effects will be. And that's really the dilemma. So although it's intriguing and exciting and interesting and probably will come, to fruition in the years to come, I think until we understand how the tool works and what it's capable of, I think it shouldn't be implemented.